Hello, everybody. This is Kevin McKernan from Medicinal Genomics. I want to cover a few key aspects of sequencing in cannabis. The recent years since 2011 have shown a few publications come out in cannabis genomics, but very few of them have actually cross-compared different laboratories' data. There was a paper by Van Beckel in 2011 that compared some medicinal genomics data to the University of Toronto data. There was a Lynch paper published maybe 2 years ago that compared some University of Colorado Boulder and Nolan Kane's data with medicinal genomics data. I think Mary Jean and Steep Hill also contributed. And why is this? Well, generally the problem we have in DNA sequencing in cannabis is the genome is in rough shape in terms of its completeness. It's probably in 100,000 pieces. Some recent longer read assemblies are getting it closer to 10,000 pieces. But it's very repetitive and it's very difficult to map in. So most people in the space aren't sequencing whole genomes. It's really only whole genome sequence that you compare one sequence to another. What's happening is people are sequencing very small panels, maybe 100,000 bases or 300,000 bases, but these bases never overlap laboratory to laboratory, and so it's very difficult to do proficiency testing to know what's going on. And this is important in a genome as complicated as cannabis, as it's easy to make mistakes. All the tools that we have for human, we need to pay very close attention to when we apply them in cannabis, because cannabis is actually much harder to sequence. I'm going to go you through, bring you through a few examples of this. This is an attempt to do, an unofficial, limited peer review in cannabis genetic sequencing, just to show people some of the pitfalls that can happen and why it's important to ask about some of these details when you're actually performing sequencing or outsourcing this stuff. So, some conflicts disclosed. We compete in the cannabis sequencing space, so clearly this is going to be a perspective that we believe in fairly strongly. But at the same time, this makes us very well-informed critics. Okay, since we do this daily and have been doing sequencing for a very long time, we actually understand this problem inside and out, more so than any critic you might face on Facebook. Okay. So what's some of our background here? Well, I worked on the Human Genome Project, managed the research and development team there at the Whitehead. I'm an author on that paper, so I have experience going all the way back to 1991. Actually, 93 is when I just started doing radioactive sequencing, but started learning fluorescent sequencing at MIT in 96. This carried on into 2000 and later, where we began reinventing the sequencing technologies. We were tired of Sanger sequencing, and so we ended up building tools, one of which was known as Solid, which is a ligation-based next-generation sequencer. This captured about 35% market share before LifeTech decided to acquire Applied Biosystems. Now, Applied Biosystems was a big company, so the $8 billion acquisition here was for their entire portfolio, but they placed a special emphasis in the acquisition announcement that they wanted to be in the next generation sequencing space, and they've done well there. I've also been involved in running the largest commercial sequencing company. Back in 2001, Agincourt Biosciences at the time was the largest commercial DNA sequencing, because Celera had actually stepped out of the space at the time and ceded that market to us. And this eventually got bought by Beckman Coulter. And in the course of Beckman Coulter buying that company, they recognized the solid sequencer needed to be its own entity, and they spun that out. And that later got acquired by ABI in an acquisition in 2006. We were also involved in building DNA sequencing and purification tools, and there's a DNA purification tool called Spry that is now probably 80% of the next generation purification market. And we have an intimate history of implementing that back at the Whitehead Institute and putting that into practice in a commercialized setting. While I was at LifeTech, I also worked on semiconductor sequencing with Jonathan Rothberg's group. This is known as Ion Torrent, and we really pushed read length as a prerogative there to complement what we were doing with Solid. Solid was very good at short reads and paired reads, and it could go very, very deep and do billions of reads, but it had a limited read length, which had some complications when you extended outside of human genomes. And then in 2011, we sequenced the first cannabis genome. We'll put it public. It's ChemDog. You can see it being referenced in John Page's wonderful paper on Purple Kush. So we have an intimate experience in this field, not only on how to use the technologies, but how to also deploy them in clinical settings where you have to be extraordinarily scrupulous about what variants you call and you have to pay very close attention to the quality of the data. And we've also been involved in actually building the tools, so we really know the bugs and the pitfalls that these machines can produce. So with that, I want to go through a bit of a technical review of some data we've been able to download from the field. And oftentimes when I do this, people in the field yell at me saying I'm being sour grapes because I'm stepping on competitors. There's nothing that could be further from the truth. The most healthy and important thing in a market is for there to be peer review, so that we actually have consumers that aren't consuming scams. And this is extraordinarily important. If anyone has seen the cannabis testing field, if there's one thing everyone agrees on, is there should be more proficiency testing. There should be more lab-to-lab comparisons so we can find out why there is variance lab-to-lab. And there is currently no PT testing or proficiency testing in the cannabis genomic space. So for all you know, you're buying garbage. And I'm going to show you some cases where you are if you're using these tools. So this market's very early. There are mistakes being made, but one thing I want to emphasize is that I do not believe any of this is done intentionally. This is actually a very hard genome to sequence, and these are pitfalls many of us have stepped in before. They're only exponentially exacerbated when you get into the cannabis genome because it is so difficult to sequence and it is so polymorphic, and you're going to see some cases of this. So most people in the field, up until we had this recent product out, were sequencing different regions of the genome. So there's a wonderful paper from I think it's the Soiler group that published, I think, 88 samples on differentiating hemp and marijuana. Great paper. There was another paper from Lynch that did like 330. There was a data dump from Phylos that was about 845 samples. I think there's been some data from Steep Hill put into NCBI as well, which is really nice PacBio data. Phylos has done some PacBio data as well to improve the reference, and I think there's even some Mary Jane data that was in the Lynch paper. So we've got about 4 or 5 groups out there that are contributing publicly to the cannabis genetic space. But up until recently, we've never really been able to cross-compare the data. We've only been able to cross-compare the whole genome sequencing data, and there's very few of those, maybe a dozen or so of those genomes out there. But the hundreds that are out there that are being utilized for certification programs, there isn't a really good way to compare them. So we set out to address this last year and built a panel that would cover at least 50 different loci from all the different data sets so we could begin to triangulate the actual data. Now, we also made the panel fairly big. Instead of it being a 50 to 100 kb panel or a 300 kb panel, you might see it's in some of the facilities out there, we built a 3 million base pair panel and then also rolled out a whole genome sequencing offering. Much more expensive, but it's you can get all the data if you want to with us. So since there is no public peer review that has actually scrutinized these methods, we think it's very important for us to take a break and look at this, see what's going on, and how effective are we at actually resequencing strains lab to lab. If we can't get that right, then anyone who's handing out certifications that this strain is yours and there's you might have some IP or you have some defensive IP, all that's out the window if the data is not reproducible. Because if it goes to court, they're going to probably ask a third-party lab, a different lab than the one who initially sequenced it, to go and sequence it again and see if they get the same answer. And if they don't, well, then you don't have anything. And so that's what we see is actually going on right now, and we thought it was important to make buyer beware. So this isn't sour grapes. This is just getting into the weeds, if you will, on the data. So here are some of the best practices that we find in clinical sequencing for sequencing humans. And this is really anchored in just making sure you can get a reproducible result. This is important for forensics. So the number one thing in next generation sequencing that uses amplified DNA, which is most of what's going on today. There are some new longer read sequencers that are very important to look at. They're a little bit noisy right now, and they're 50-fold more expensive, but they don't use amplified DNA. The really cheap, fast sequencers right now from Illumina and Ion Torrent, they all use amplified DNA. There are some PCR-free methods you can use, difficult to use them on plants with limited amount of DNA. And so what you find is people are amplifying their DNA libraries. When you do that, you have to make sure you have a method to remove PCR duplicates because you want to be able to discern from PCR selectively amplifying one allele over the other. And the only way to do that is have a method to remove PCR duplicates. Sometimes people use molecular barcodes to do this. Other times, the nature of the data reveals where the duplicates are. In other words, When you see pileups of reads, those are generally, that all have the same start and stop point, you assume that those come from the same molecule. If you have coverage that has reads that start and stop at different points, you can assume those are all novel molecules that were sequenced, and you can treat them as such. And that's very important for counting alleles. We're going to touch on that. Then there's a longer reads issue. We need to have close attention to read length when you're doing DNA sequencing. This is really important in cannabis genomics because the shorter the reads, the less unique they are, the less accurately you can place them in the genome, and the more easily you confuse them and put them in the wrong place. And if you start putting them in the wrong place, you start calling SNPs that don't exist. So longer reads are very, very critical. We're going to touch on what lengths are required to really either to handle human genomes, and you need even longer ones to handle cannabis. Paired reads are important. What are paired reads? Well, when you read one end of a DNA molecule, if that one isn't perfectly unique, you have nothing that you can do with it than place it randomly to one of the many places it maps, or to throw it out, or you rely on a read coming from the other direction on the strand, which might be 100 or 1,000 base pairs away. These are paired reads which have much higher signature in placing them in the genome. You can be far more confident in a SNP call if you know it's come from a paired read, and many algorithms actually do this. They do not make any SNP calls unless the reads are paired, because only those do they believe are anchored in the genome in the right place. So paired reads are very, very important. The other thing people will do, if they're using a PCR-based approach that is that's using human-designed primers to amplify regions of the genome, as opposed to universal primers you might find when you shear DNA and just glue them on. There's two very different approaches, and we're going to touch on why those matter. These human-designed primers have a design bias, that you design them according to the reference genome that you're using. And if there happen to be SNPs in those primer sequences, well, then you tend to amplify only one allele and you miss the other. So to get around this, most clinical laboratories, if they're taking an amplicon-based approach, will tile multiple amplicons across the variants that they want to look at, so that in case some of those primers land on SNPs and allele drops out, they'll catch it with another set of primers. So it's important, if you take this type of human-designed primer approach, that you have multiple overlapping amplicons that cover the SNP. Otherwise, you don't have confidence that you've captured both alleles. This is very, very critical, and it's sequencing 101. If you don't do this, your data will be thrown out. Preferably use sheared DNA if you can. And the reason for this is that sheared DNA gives you random start and stop points with molecules. And those random start and stop points inform you that the molecule is in fact unique. When they all line up like they do with PCR, you can't discern whether it was a PCR amplification artifact or whether you're actually surveying both alleles in the genome. Critically important, most clinical sequencing today is done with SureSelect, which is a system that uses enzymes to digest the DNA and then glues on universal primers such that you get random amplicons that cover the entire genome. You get much better coverage consensus this way, much more accurate results. It's what is done clinically to call humans, at least a clinical human disease. Okay, so the other thing to avoid if you're going to make amplicons is to avoid very short ones. If you make very short amplicons, the problem you run into is that the PCR primers that you design are usually about 25 bases on each end. And if what you're targeting is only 50 bases, well, then you have 50 bases of primers and 50 bases of target. That is going to lead you with a tremendous amount of primer bias, where more of your amplicon is prone to SNPs being under your primers than the actual target you're trying to sequence. This is a horrendous way to guarantee that you will have allele imbalance, and your heterozygosity information will be thrown off. Okay, so let's talk about how people handle these PCR duplications. So the Broad Institute is one of the premier facilities for doing DNA sequencing, and they wrote a program called Picard. And Picard is something that goes through and scans for PCR duplication events and removes them. Okay, so here's an example of, you've got reads that are starting in the green are going in one direction, the red are going in the other direction, but they start and they stop at defined locations. So you know each of these were derived from a unique molecule. However, when you get here, they all have the same start and stop. And so now you don't know if that's just PCR took off on that one particular amplicon and over-replicated it because it was selectively amplified due to its sequence context, or whether that is actually a real representation of the native genome. So these, it instantly takes and removes. In fact, this is standard in clinical sequencing, is that any data that looks like this, you toss it all out, because you can have Darwinian amplification, where something based on the nature of this PCR product, maybe a SNP in there or something else, makes this piece of DNA amplify faster than this one. And then bang, these take off, and you don't know if you're really sampling both alleles. Very important the DNA levels are low as well. There's other places you can go to look at this. There's a lot of peer-reviewed publications that discuss what happens when this is present, what happens when it's gone. But generally what happens is you lose heterozygosity. You miss one of the alleles, or you can't properly quantitate copy number variation, and so your variant calling gets all screwed up. So this is important to know, because what we're seeing in a lot of the data that's out in the cannabis industry right now is that it looks like this. They are designing PCR products that they're sequencing, and in any professional genome center, all of this data would be thrown out. It would never be utilized, yet this is being sold to customers in the cannabis space. Second issue that's going on with this data is they're using incredibly short reads. In fact, this standard is probably 10 years old. These are 75 base pair reads, and they're only unidirectional. You'll see they're all going one direction here. So they're taking a PCR product, a short one, 100 base pair PCR product, and they're cloning it. And when I say cloning it, they're adding primers on the ends of that DNA that match the Illumina sequencer. When you do that, the DNA molecule can glue in the forward orientation or the reverse orientation. So you get both directionalities out of the molecule, but your reads are only coming off the forward primer. So this is your primer sequence here. And their reads the Illumina primer that's on the array is right here. It's tailed, you can't see it. It sits here, sequences across your primer sequence. This is all man designed, so this isn't real sequencing variation that's going on here. A lot of people get confused by that when they design primers. They don't realize that their primer sequence cannot be genotyped because it's not real, it's what you designed. And then this 75 base pair read gets in about 50 bases into the insert before it stops. And you get the same coverage going this direction because they're reading it from the opposite direction when the molecule gets cloned into the opposite orientation. So you get double coverage in the middle, and then you run, and then it stops before it hits the other primer sequence. So this gives you this kind of top hat coverage. Why is this messy? Well, this is what's actually happening when you sequence this with something like SureSelect. You can see in this case, these people designed the primers where the 3' end match their reference. And that meant it could only amplify that allele, and it failed to amplify these alleles. So you can see here, these are much longer reads. They're paired 150 mers. This is what we run at medicinal genomics off of a SureSelect. So we have random start points on DNA molecules that cover this area. They have reads over here that are 150 base pairs long, so they're much better to map than these tiny 75 mers. And another 150 base pair read over here. And all of the read is actually genomic DNA, as opposed to here we have half of the DNA sequence is synthetic, which you have to throw out. And as a result, they're making homozygous calls when the data is clearly heterozygous. And so this has this sample misgenotyped. And if anyone goes to recheck this, even after they've been given a certification, they're going to find that the sample was genotyped wrong, and they don't actually know what they have. And so their certification is bogus. So this is critical to really dig into. So let's expand on this a little bit. So what is going on when you have this much primer territory in what you are trying to sequence? And what I mean by this much primer territory, this is 25 bases here, this is 25 bases here, and there's 50 in between. So already you've got half of your DNA sequence is synthetic, human designed, and that means it could fall on top of an unknown variant that's in the genome and selectively amplify only one allele. Okay, so we are seeing some of that right here. This right on the 3' end of the primer, it's only amplifying this one allele, and we're ending up with marginalized data. the more primer space that you have, for instance, if they made this a single base in between, you would have a 50-to-1 ratio of primers to novel DNA that you're going to query, which means you're going to run into a lot of SNPs that throw off your assay. Now, in cannabis, I mean, this is a big no-no to do in the human genome. There's a SNP every 1,000 bases in the human genome. The cannabis, there's a SNP every 100 bases, which means the likelihood of there being a SNP under your primer is like 50% if you have 50 base pair primers like this. All right, if you only have a single base in here and you have to keep tiling these primers, your likelihood of having a SNP underneath your primers just goes up. So now, there's a SNP every 100 bases in cannabis genome-wide. When you get involved into the regions that are under selection, like THC synthase and CBD synthase and the terpene synthase pathways, the polymorphism rate shoots up to like 1 every 30 bases. And so you're almost guaranteed to have a SNP under your primer. So if you take an approach where you're designing primers like this, you're always going to have allele bias. It's just a horrendous idea. We know this because we played around with a technique called RADseq, which has similar setbacks. The first version of StrainSeq we had used restriction enzymes. Restriction enzymes use 4 or 6 base pair regions, and there's a likelihood that you can get SNPs in those and get what are known as RIFLIPs. And so it has a similar problem, albeit a little bit less severe as PCR, but it still leaves you with these universal start and stop points, which means you have a hard time counting which molecules are duplicated and which ones aren't. So we abandoned that approach because we recognized it was giving us distorted allele ratios. You can Google this, allele dropout in PCR. There's hundreds of papers on this. This is a known problem in clinical sequencing in humans. This problem is 10-fold to 30-fold more problematic in cannabis, and so you really cannot afford to take approaches like this. Otherwise, you're going to have absolutely bogus data that no one else can reproduce. So the other thing to consider on this, as I mentioned before, these bases, which are your primers, this is not real sequence data. You have to throw this out. You can't call any of this stuff in here because this was designed by your oligosynthesizer, and your polymerase saw this union hybridize on the genome and only started incorporating bases in here. So only these ones really reflect what was on the reverse complement strand of the polymerized DNA sequence. Likewise, when you run into this direction, you start hitting the reverse primer, this data should also be thrown out. So in here, 50% of the data delivering is thrown out, and the 50% of the data in the middle is marginalized because it was derived by primers that potentially could sit on SNPs, and you may be only looking at one allele. Here you can see there's a couple other alleles that are showing up, but sometimes 3. And the reason why you sometimes see 3 alleles is usually a sign of mismapping. You're in a region that might have a copy number variation somewhere else in the genome, and so you start calling 3 alleles. This is a diploid plant. There should only be 2. And when there are 3, you're not mapping your reads correctly. And sometimes there's nothing you can do about that when you're dealing with short reads. So what is this primer bias all about? Well, it really comes down to is the polymerases that copy DNA are extraordinarily sensitive on the 3' end and making sure these bases match before they extend and copy DNA. If they don't match, it doesn't extend. And so what happens is if you have any SNPs that are in your primers that you weren't anticipating because you designed them off of, let's say, a canatonic reference or something else, if you have that present, then your primer's not going to hybridize. You're not going to amplify that allele. You might amplify something that matches what you anticipated, but then you're only getting recapitulated the information back that you already knew. And so this distorts the entire genotyping process towards the reference. Okay, it distorts the heterozygosity calculations. It distorts the phylogenetics. The phylogenetic trees become a hallucination that are all bent towards the reference you designed the primers off of. And it's never very reproducible because this artifact is very, very TM and salt sensitive. And if you have a different DNA prep that has slightly more or less salt, these mismatches are going to behave in a different way. And they're going to give you really erratic reproducibility. This problem gets worse with higher polymorphism rate. So as we go from human to cannabis, which the polymorphism rate goes up 10-fold, you can't find a place to put primers that won't do this. So this is not a certification because the genotyping is just dead wrong. It's worthless for IP because it can't be reproduced and it doesn't actually represent the real background and the real variation that's going on in the plant. Okay, so coverage uniformity is important. And what is this? Well, you can see you have this kind of top hat coverage mechanism that's going on here in looking at this data. And when this occurs, we mentioned before, you get certain artifacts. Like you can see here, there's an artifact at the end of the DNA sequencing. Okay, this is what's known as a +A artifact. When polymerases hit the end of a DNA molecule, they sometimes tack on an A, and they do it so reproducibly that this is actually a mechanism that Illumina uses to clone their DNA. Occasionally they add 2 A's, and occasionally this gets genotyped incorrectly. What you really want to have is something that's far more uniform in coverage. Okay, this is the coverage curve right up here. We designed this amplicon or this sure select region to tile 50 of the loci that are in this other company's sequencing panel so that we could triangulate anything we sequence against their database. And most often when we do that, we always find there's a couple SNPs that they miscalled. So the average person getting sequencing done there is getting wrong genotypes, and they don't really have a certification that they're being sold. Now what you can see in this data as well is that you have alternate start points. You have molecules starting in different places, so you know they're unique. They were not PCR artifacts, and they are much longer. They are closer to, these are probably 150 or 125 base pair reads, and they're paired, meaning there's a forward read and there's a reverse read. And this means if this one has difficult mappability because there's an insertion or deletion in it, you can anchor the thing off of this one and properly map this and know this one goes right next door. Paired reads is utterly critical when it comes to mappability in complex genomes. So as I mentioned, any professional genome center tosses all this data out. It's complete garbage. So read length, what we're seeing, a lot of these folks are cutting corners on cost. They're only running 75 mers. 75 mers was in vogue in 2008, about 10 years ago, but almost all systems today sequence 150 mers or more, and we even have some long read systems coming out that sequence 10 kb and 100 kb. So increasing read length is in vogue. It's more expensive right now. These longer read systems are often 10 to 50 times more expensive than Illumina, and they have some error rates that need to be known and adjusted for. But they're the next wave that are going to be coming once they're affordable enough and once they're cheap enough. It's very difficult, though, however, to run something in the couple $100 range with these platforms and still have all the coverage and all the SNPs that you want. Right now, though, there's an asymptotic curve on read length that is really important to underscore. The difference between 75 mers and 150 mers is quite stunning, and I'm going to show you that in a minute. And then the other thing we want to emphasize is primer-free methods are really are critical. You have to use methods that shear the DNA or digest the DNA and then glue on the primers with ligases. If you don't do that, you end up having a bias in your design of your primers. You design it toward one reference, and that means everything you do genotyping with those is going to bend everything in the phylogenetic tree toward your primer design. It should be avoided. So, short read mappability in humans. This is an easy problem in humans, but what you can see here is when the read length goes from 50 bases, like we're seeing with some of these panels, there is this steep curve of mappability where the positions within the genome that can be uniquely mapped. And you can see as you get out to 100 and 150, this begins to asymptote. That's where you want to be for sequencing in human. And you can see what this mappability looks like if you go to the UCSC Human Genome Browser. You can click on a tab that says, show me the mappability of these regions with 24 mers, 36 mers, 40 mers, 50 mers, 75 mers, 100 mers. And you can see as you get out 100 mers, this begins to all fill in. But if you're operating in this space, you have all these zones where you don't know how to map. And your reads could fit there, but they could also fit here. And so you start getting confused as to where the SNP's really coming from. In cannabis, this problem is far worse because the genome's in many pieces. It's 64% AT, which means it's also highly repetitive, which means the mappability it requires even longer reads. Hence, you should use paired reads if you can. So just to reiterate, building phylogenetic trees with primer bias, this means the entire tree is built on allele dropout and will bend all of the strains toward the reference strain due to the primer design selectively amplifying the expected sequences. And as you can see in many other publications online, this type of data is always thrown out. It's not utilized, yet it's getting utilized in cheap genetic tests in cannabis because people aren't asking these detailed questions about, hey, how's the sequencing done? How many SNPs do you have? What's the target region? What's your coverage uniformity? These are all questions that get asked in the clinical sequencing space, as you really need to understand these things to know what type of reproducibility the laboratory has. So in conclusions, the genomic cannabis genomic certifications, they really need to utilize best practices for genotyping by sequencing, or labs won't be able to reproduce your fingerprint, and then you don't really have one. PCR-based approaches should be avoided. They just, in cannabis that is this polymorphic, you cannot do it successfully. You need probably databases of tens of thousands of strains fully genome sequenced, which we don't really have right now. If any genome sequence is done, it's done and mapped to somewhat questionable references. And you would need that information to know where every single SNP is in the population, so you don't drop a primer on them. That is no one's going to hold their breath for that perfect reference. The right thing to do is to just use technologies that don't rely on primer design. This is what is standard in SureSelect, and is the vast majority of people in the next-gen sequencing space are using that platform because of this problem. This is 10x more important in cannabis because the polymorphism rate is tenfold higher. You're going to have SNPs under primers in a tenfold more frequent rate in cannabis than you are going to have them in human. And even in humans, when they're 1 every 1,000 bases, people avoid doing primer design like this like the plague, because it just leads them down avenues where they're not even aware that they made the mistake. They just see higher polymorphism rates or higher homozygosity rates. So longer reads and pair reads should be employed. You need to enhance the mappability, otherwise you're going to be piling on repeats on top of each other. This cannot be underscored enough when the reads are highly polymorphic. When you add that much entropy to the DNA sequence information, you begin to have to look and scan to place the reads in more places. This is like trying to put a puzzle together where a lot of the puzzle pieces are constantly blurring on you. You have a hard time locking them in place when you have to consider much more entropy in the sequence in order to actually map them. If you have to allow reads to map with 4 mismatches, which sometimes you do in cannabis, you can place them in a dozen more places in the genome when you allow for that many mismatches to occur in the sequence to put it somewhere. Okay, so we have to have the capacity to do that in this genome because it is that polymorphic. So the only way to really deal with this is with paired reads. You can't really do this with 75 mers. And then the other questions people should be asking is, what's the total target area that we're sequencing? If I'm having a $300 or $500 test, what am I getting for that? Am I getting 300,000 bases, or am I getting 3 million bases? This is kind of a price per gig question that is a standard in DNA sequencing. You should also know the number of SNPs that are coming out of that report. If it's 2 SNPs, well, you know what? You don't really have a report. If it's 1,000 SNPs, interesting, you can probably fingerprint with that. You want to know that the SNPs are called correctly, but you can't really use that for scanning for phenotype to genotype correlations, because that's only 1 marker every 10 markers every chromosome or something. Well, there's 20 chromosomes in cannabis, plus a chloroplast, plus a mitogenome. So when you lay 1,000 SNPs across that, you don't have a lot of SNPs per chromosome. You definitely don't have enough SNPs to find phenotypes. Selling this type of data without this detail in many other fields is considered a scam. You need to. This is like transparency. What am I buying? So it's important to ask those questions if they're not providing them. And then phylogenetics based on garbage data is a mirage. It's there, it makes nice pictures, but if the underlying data has a primer design problem, and you're dealing with SNPs like this, you're not really creating a phylogenetic tree that's meaningful. You're creating one that's all getting bent toward the reference genome. So, and finally, I would just say DNA sequencing techniques are always evolving. People do go up and update their databases. They refilter their SNPs to try and improve the relationships that they can get. And so you really want to have a tamper-evident database so that when your database changes due to new software upgrades or new filtering of the data, that your certification doesn't get changed in the process. You need a timestamp as to when it was sequenced and what the genotype calls were at that time. If you're sitting in a database where they are refactoring the software and the SNP calling algorithms to try to dance around some of these problems and refine them, well, it doesn't help you if you don't actually have a record of the original genotype that you recorded at the earliest date and time. You really want to have databases that can demonstrate that it is tamper evident. So we do this with the Dash blockchain. Whenever we sequence, we put our VCF files into the Dash blockchain, and that gives us and our customers confidence that the version 1 sequence on the first timestamp is X, and if you ever change that, there's evidence of it. The hash changes. So this is incredibly important when it comes to certification. So hopefully that has been helpful to you, and that's just a quick tour through DNA sequencing with next generation tools and mappability. But just keep in mind, garbage in is garbage out, and I think that's pretty much what's going on in the field right now with a lot of the techniques out there. So thank you for your time.